This article, titled Microbiota-gut-brain axis multi-organ chip construction and applications in drug evaluation, was published in iMetaOmics. I, Tang, am the first author of this article, and I will now introduce it. Human gut microbiota is symbiotic with the host. Its dysbiosis is linked to multiple diseases. Via neural, endocrine, immune, and metabolic pathways, microbiota engages in bidirectional communication with the brain through the microbiota-gut-brain axis. Traditional research models are struggle to replicate the human body's complex physiological environment. Microfluidic organ-on-a-chip technology dynamically mimics the microscale physiological environment of human organs, offering a platform for complex disease study and drug development. The highlight of this review lies in the systematic integration of the MGBA and multi-organ chip technology, and the comprehensive exposition of the development status and application significance of each module unit in the system in the form of technological iteration, demonstrating the key application value of MGBA multi-organ chips in disease research and new drug development. The MGBA is structured around 4 systemic pathways and 2 physiological barriers. Vagal afferent and efferent fibers establish the most direct neural pathway linking the gut and brain. The endocrine pathway, governed by the hypothalamic pituitary adrenal axis, mediates stress responses. Its dysregulation increases glucocorticoid secretion, altering gut microbiota composition and metabolic activity. Gut dysbiosis triggers the release of pro-inflammatory mediators, which disrupt intestinal and blood-brain barrier permeability to induce neuroinflammation. Microbial metabolites enter the circulatory/lymphatic system or directly act on vagal/spinal afferent neurons, mediating neuroregulation, immune signaling, and blood-brain barrier remodeling. Current gut-on-a-chip mainly relies on synergistic effects of fluid shear stress and mechanical peristalsis to promote intestinal barrier formation. For deeper host-microbe interaction studies, luminal anaerobic gradient construction is a key challenge, with current methods including establishing hypoxia via nitrogen flushing, naturally reducing oxygen via permeable membranes combined with computer modeling, or oxygen regulation through time difference stages, with real-time monitoring via oxygen sensors. As model advances, applications expand to simulating depression-related gut pathology and, with machine learning, enable studies on various intestinal diseases, drug development, and probiotic screening. Blood-brain barrier on a chip features vascular and neural compartments separated by a porous membrane, with endothelial cells and tight junctions forming the core barrier structure. Fluid shear stress mimics in vivo hemodynamics to promote 3D vascular-like structure formation. This model enables central nervous system targeted drug design and assessment of transbarrier efficacy or therapeutic effects of active components. Disease-specific models could further advance personalized medicine. Brain-on-a-chip features neuron-glial co-culture, synaptogenesis, intercellular communication, and immune simulation. Using 3D hydrogels and microfluidics to mimic bidirectional cerebrospinal fluid perfusion, it replicates neural networks and brain morphology. Combined with in situ induced pluripotent stem cells differentiation, it generates human brain organoids. Astrocyte neuron co-culture studies also verify synaptogenesis and spontaneous neural activity in the chip. In disease research, it recapitulates pathologies of neurological disorders and investigates external environmental impacts on neurodevelopmental damage. Multi-organ system interconnects organ-on-a-chips via microfluidic channels to study cross-organ interactions. Multi-organ systems fall into 2 types, single-organ chip coupling system and multi-organ on-a-chip plates. The core of system interconnection is constructing physiological vascular networks, with current methods including 3D bioprinted large-scale vectorized tissues and microchannel formation via endothelial cells. Based on interconnection modes, system is classified into static, semi-static, and flexible cascade systems. In disease research, system offers a human physiologically relevant platform. Gut-blood-brain barrier-brain chip integration research advances, with the European Research Council funding the pioneering Minerva Bionic platform in 2017. And research reports on split-type microbiota-gut-brain axis chips. However, current systems remain technologically immature, facing bottlenecks such as material adsorption interference, heterogeneous microenvironment demands for co-cultured cells, difficulty in long-term cell viability maintenance, limited capability for dynamic signal monitoring and imbalanced microfluidic control across organs. Microbiota-gut-brain axis multi-organ chip systems, leveraging biomimetic dynamic microenvironments, enable neurological disease mechanism analysis, drug evaluation, and gut-brain crosstalk elucidation, advancing gut science frontiers.